Hi guys, hope you all are well. Welcome to my channel Shrutapa's Biology. I am Shrutapa Datta. Now I shall continue the topic biomolecules, and in biomolecules in my last video I have told you that DNA and RNA, that is the nucleic acids, those are also included in the biomolecules. In class 12 when you will study the genetics, then you will get more illustrated idea about these things, but now I am giving you some basic idea about the structure of the Washcentric model of DNA, and that is the main model of DNA, and another is different types of RNA in short. Okay, so let's start the topic. Watson-Crick's model and Crick's model of VDNA. It's very important for your board exam, as in 4 marks you will get this question, and all the details of this is very important for the competitive exam. So just watch the full video with full attention. It's not a very large video, or this is not long video. It's a very short description, but all informations are very much important. Okay, so number one is, firstly I am drawing the structure of the Washcentric model. In all the book you will get the diagram, but I am giving a very rough diagram of it. This is the imaginary axis, central axis. On surrounding imaginary axis, but like surrounding this, the two polynucleotide chains are wound around, and in right hand, that is in clockwise manner, it's wound around. They have their line on the sugar phosphate backbone. Already in my last video I have told you that's the pin to sugar. So 5 carbon chains are there. 5 carbon ring is there. Naturally is the pentagon. So this is the sugar phosphate backbone, and here the nitrogenous bases are present in this manner. A, T, G, C, T, A, in this manner. They are linked with each other by hydrogen bond. Adenine is attached with thymine. In between this, there are 5 base pairs. And another thing is very important, that is the position of the 2 polynucleotide chain. That is, if this is the 3' end, then another one is 5'. So they are anti-parallelly coiled with each other. So in here it will be the 3' and 5' ends, they are facing each other on the top and at the bottom also. At the top and bottom, in both the places, the 3' and 5' faces, 5' ends are facing each other. Now another thing is that the distance between 2 base pairs, suppose this base pair and this base pair, among them the distance is known as distance is measured as 3.4 angstrom. So naturally, if in between these two 3.4, then in the single turn there are 10 base pairs. I have already told you. So 3.4 into 10, so 34 angstrom. This should be 34 angstrom. 3.4 into 10. Now the next thing is that this is the complete turn where one minor group and one major group will be there. This is known as minor group. And this one, the shallow one is known as minor group and the deep one is known as major group. So in one turn, there should be one major group and one minor group. Another thing is that in between these two polynucleotide chain, the measurement is 20 angstrom. So the diameter of the DNA is 20 angstrom. These are the facts here. And another thing is there that in between the adenine and thymine, there is double hydrogen bond, and in between cytosine and guanine, there is triple hydrogen bond. So A double bond T and C triple bond G or G triple bond C. This is about this. And another thing is that after attaining all these characters, it's known as the physical structure of DNA also. Washcentric's model, as well as it's known as physical structure of DNA. In question you can get anyone, either physical structure of DNA or Washcentric model, both the same. And all the informations of it, that is all the points, each and every point you can get in competitive exam in MCQs. Like what's the diameter of the bDNA, or what's the diameter of the Washcentric's model, and what is the distance between 2 base pairs, etc. All these MCQs can come. And now I am telling you all the points again, starting from the first one. 2 polynucleotide chains are helically coiled with each other in antiparallel manner. What is antiparallel? This is antiparallel. The cap of the marker here and here another cap. Both the caps are not at the same side. So what's the significance of it? Why it's present in this manner? As both the if they remain in parallel manner, not in anti-parallel, suppose both the caps are on the same side, then they will not match with, they will not make pair. As adenine can't make pair with adenine, adenine can make pair with thymine only. So in this manner it will go on. So that's why it's this is the one chain is complementary to other. Suppose this is one polynucleotide chain, this one is complementary to this, or this one is complementary to this. Okay, so number one point is polynucleotide chains are coiled with each other in anti-parallel manner, and one is complementary to other. Number two, the diameter of the DNA is 20 angstrom. Number three, the coils are on around the central axis in right-handed manner. Number four, the distance between two base pairs is 3.4 angstrom. Number five, in one turn of DNA, there should be one major groove and another minor groove. Next point, in one turn of DNA, there are 10 base pairs. So 3.4 angstrom into 10 is 34 angstrom. So the total length is 34 angstrom of one turn. In a single turn, there should be the one major groove and another minor groove. And next one is that the nitrogenous bases are lying on the sugar phosphate backbone, and in between two nitrogenous bases, there are hydrogen bonding, and this hydrogen bond is in between A and T, that is adenine and thymine, there double bond, and guanine and cytosine, that is triple bond. So we know that the one purine is making bond with another pyrimidine. In AT, A is the purine and T is the pyrimidine. In CG, C is the pyrimidine and G is the purine. And next is that the Chargaff's rule. According to Chargaff's rule, A plus G is equal to C plus T. That means in a DNA, total amount of the purine and total amount of the pyrimidine is same. That is in 1:1. This is the Chargaff's rule. So this is about the structure of DNA. After that, I am coming to the structure of RNAs. First, I am telling the structure of RNA is not so important. Just the informations are important, short informations like that. There are RNA always single stranded. RNA has no double strand. RNA is always single stranded. And another thing is that in case of RNA, they just help the assist the DNA in function. They don't have any direct function, rather they have the assistant functions of like the assistant of DNA. How? I am telling this. There are three types of RNAs. Types of RNA, that is the RNA world. Number one, mRNA or messenger RNA. Number two, tRNA or transfer RNA. And number three is rRNA or ribosomal RNA. Three types of RNAs are there. Now, what's the function of the messenger RNA? The function of the messenger RNA is it just acts as the messenger of DNA. Like during protein synthesis, the DNA makes the copy of the messenger RNA while it just writes the informations, and mRNA takes that code and come to the cytoplasm to perform the rest part, that is the translation. That's the last part of the protein synthesis. So the function of the mRNA is mostly is that, and the newly synthesized mRNA is known as HNRNA, heterogeneous RNA. Heterogeneous RNA is nascent RNA. And mRNA is always linear in structure. It's single stranded and linear in structure. And number 2 is tRNA. tRNA is it's also a single stranded structure, and it has the function to bear the amino acid at the place of protein synthesis. So they help in the formation of the polypeptide chain by bringing the amino acids to the site of protein synthesis. And they have, during their protein synthesis, when they help in that procedure, then they fold in such a manner, then they look like the double bonded structure, and that's known as clover leaf model. So tRNA during functional form, it has the clover leaf model. That means clover leaf, you know, clover I think लंग का जो पत्ता होता है ना, उसके जैसा ये फोल्ड करके स्ट्रक्चर लेते हैं. इसमें 4 साइड्स रहता है, दैट एंटीकोडन लूप, DHO loop, CC amino acid binding loop, and another is T5C loop. So different loops are there, and this helps in the protein synthesis by carrying the amino acids. And the function of the rRNA is it forms the ribosome, and most abundant RNA is rRNA. rRNA is most abundant RNA. As in all ribosomes, rRNAs are there, and all the ribosomes are made up of many rRNAs, and ribosomes in a cell are numerous in number. So naturally, the abundant of the most abundant RNA is rRNA. So this is about the different structure of the nucleic acid, and this is the last of this biomolecule. So bye today.